Good afternoon, everyone, and welcome to Yahara Live. I'm very excited about today's talk, and I hope you are too. Now, if you've attended a Yahara Live in the past, you've probably heard from Dr. James Smagala, but today we're giving him the spotlight to talk about an amazing example of how AI and machine learning tools can be implemented into real important research. So without further ado, James, feel free to take it away.
Austin, thank you for the introduction. And to everybody joining us today, thank you. Welcome. This is a bit of a repeat of a talk that I gave at BioIT about a month ago. And it's the first time I've gotten to do some novel research in a little while, so I'm excited to share some of that with you. We'll take a little bit deeper dive than I did during that session into some of the machine learning aspects of this. And if you've got questions along the way, please do feel free to ask in chat. So, to start us off a little bit, where I'd like to go today is looking at leveraging some more novel machine learning techniques to directly detect things that have been difficult to see in other circumstances. And I'm going to frame this in terms of the SARS-CoV-2 pandemic because I happen to have really good data from that time period. But this applies much more generically across a number of bioinformatics and machine learning applications. So there should be something applicable out of this if you're interested in those domains at all. If I could give you a 3-month head start on the next pandemic, would you take it? Effectively, we had that during the SARS-CoV-2 pandemic. We knew that this was a serious problem in China, and the world generally didn't know what to do about it, and we didn't know what virus was causing it. Um, but it's possible we should have had an even longer head start on this. If you take a look at the timeline, we actually start locking down a number of different parts of society starting about March 11th, 2020. If you work backwards on that time frame, we had full genome sequence of the SARS-CoV-2 virus first part of January. So we knew 3 months in advance. We knew that there was significant number of patients in China experiencing an atypical pneumonia. And it's possible that if we had had the wherewithal to be looking for it, that we could have seen SARS-CoV-2 in wastewater samples in Spain a full year in front of the actual outbreak. Now, that study's been questioned. Could it be contamination? Yes. But it was almost certainly circulating at some level in the human population well prior to the December 2019 outbreak of the atypical pneumonias in China. So it's hard to tell exactly how much of a head start we could have gotten, but we need to go look for these kinds of threats if we are going to be able to detect them and decide what to do about them early. Pre pre Deploy our full use of preventative technologies and quarantine procedures and outbreak controls and anything else in the public health domain. So there's a emerging project that's coming from the US government right at the moment that says we would like to do a broader biothreat radar surveillance system. So we're going to look at more metagenomic kinds of sequencing. We are going to look at a wider variety of sampling methods. So we're going to take air samples and wastewater samples and do pooled travelers genomic surveillance as people are coming off of airplanes and a number of these things, and then be able to detect known pathogens. But there's a secondary piece of it that says, and we should be looking for the unknown ones. That's a little harder topic to address. It's easy to find the things you know about. It's harder to find the things you don't know about yet. So let's talk a little bit about some of the downstream bioinformatics problems that you run into here. We have the capability to do full-blown de novo assembly So start from just a bunch of raw reads and see if you can assemble a meaningful bunch of those together into a novel pathogen. This is very expensive. It does not scale very well, and it takes a long, long time. So mostly we don't do it, not at scale. You can do it in specialty cases where you know you are trying to detect something, but right at the moment we don't have a way to do this on the kinds of scales that we're talking about for a national scale surveillance program. There's a heavy database dependency in here too. Um, this works far better if you know what it is that you are trying to find, and so you end up looking for a pathogen that's similar to ones that you have known in the past, and that requires that your reference database be very good, or that you be updating it very consistently over time. Again, relatively difficult. There is sort of a hybrid assembly approach in the middle of this, and that's what we're going to pick apart and use a little bit today. The CDC previously put together the Iterative Refinement Meta Assembler, IRMA, which does a pretty good job of capturing novel viruses as long as they are close enough to a reference. It's not de novo assembly, it's fast enough to use at scale, but it does not work well if you have very distantly related pathogens. And that's what we're going to lean on a little bit today. So, the framing question. Can we detect the SARS-CoV-2 emerging reference strain? We're pretending it's pre-2020. Can we detect an emerging virus using a really bad reference? There are other coronaviruses that circulate. There are several that show up, at least three different kinds that show up as part of the common cold. And there have been a couple other crossovers, the original SARS-CoV-1 and MERS, which tends to come out of camels, cross over into humans, and have a relatively high fatality rate, but it doesn't spread very well. So you probably haven't heard about that in many years. We're going to pick that MERS reference and ask, can we use a bad reference to find a virus? And this is the simulation for, I've never seen this virus before, but it's like something or close enough to it in the database. So, our SARS-CoV-2 and MERS have less than 50% protein sequence similarity. This is very, very divergent, but they are both coronaviruses. And the intuition here is the structure of the way the proteins fold has to stay relatively consistent in order to get functional proteins and infectious virus particles back out of it. So there should be some kind of similarity here, if only we could figure out where to look for it. We're going to back that intuition up with some recent research from the Bloom Laboratory. They were working on influenza HA subtypes. So they're looking at H3, H5, and H7 HA um, proteins, noticing that they have significant divergence between them, very low amino acid identity percentages, and yet the alignments between these is within a couple of nanometers for the structural representation of the protein. The structures overlap heavily, even though the amino acids do not. That's the confirmation that what we need to be doing is looking in a higher structural space. The amazing revolution here in the last few years has been originally AlphaFold from Google. Um, we don't have access to that for commercial use, so we're going to substitute in a few other ideas here, and that's where we start to ramp in some of our machine learning. I also want to take just a minute and talk about IRMA. I mentioned it earlier. It is the iterative refinement meta assembler, so we're going to start with a template that looks kind of like the virus we're trying to find, match to it, and then iterate and refine the reference until we have pulled all the new reads out, and by the end of it, we should have one new virus. So that iterative idea of looking for things that I can match and then adding additional things as my reference improves. So here is the follow-up to the AlphaFold comment. There is another protein language model competitor out there that is achieving some very similar and in some cases better set of results. This is the ESM family of models. So these are trained on the raw sequence, the structural elements that live above that, the way that the protein starts to fold. And then there's even sort of a third level that you can translate that into structure encoding alphabets. And for reasons of keeping it nicely compatible with the existing set of amino acid tooling, we're going to go ahead and pick 20 characters. But those 20 characters in our structure alphabet are not the same thing as a amino acid. These are now structure encoding characters, and they tend to be far more divergent than amino acids. So an amino acid may slot in several places in the structure. The structure encoding characters tend to be much more specific about exactly which structural elements of a folded protein they will associate with. We're going to use the version 2 models because that's the code that I started with when I started working on this. The version 3 models are out, and they're about twice as efficient at doing these kinds of structure encodings. The very best of the ESM3 family that is out right now is competitive or better than the AlphaFold family right at the moment. So if you need access to high-end machine learning for protein structures or other kinds that might fit better in your laboratory situation, stop by and talk to us. This is stuff that we can set you up to be able to host and use as you go forward. Um, what are we going to do with that? What we're going to do with that is encode our raw reads and just look for um, encoded proteins that have, or out of the DNA, encode up into structural space and look for structural characters that have overlap with the MERS genome. And if we do that, it takes a number of iterations, but you can completely recover the SARS-CoV-2 assembly to 98%. We're missing a handful of reads out at the tail end from reads where we have almost no nucleic acid overlap and less than 50% amino acid overlap. And if we take a look at when this what's going on here, I am working on the original Wuhan strain, a metagenomic sequencing of it, and no reads, none, map to the MERS genome. If you do them with translated BLAST, you find proteins where you get almost no overlap. You find a few where you do get some. These are highly conserved. If we now smooth that out with a structure encoding alphabet, all of a sudden I'm recovering 80 to 85% of the reads in the first pass because they do map to their similar protein structures, even in a highly divergent virus. So here are different SARS-CoV-2 genes, and you can see the tier 2 is just using MMSeq2 to simulate a translated BLAST. But the tier 3 is where we've gone to structure encoding, and you can see cases where the structure encoding wildly outperforms what you can get from the little bit of amino acid coverage that you might get. That kind of novel approach means that now my reference database is much easier to maintain. As long as I have the right set of references in it, I can do this structure encoding trick and not just search against SARS-CoV-2, but against a known set of viral reads or bacterial reads and see, is there a structural overlap with a pathogen, with an organism that I already understand, even though I do not know anything about this emerging threat in my sample? That approach does scale. because now it applies across a whole bunch of different potential reference genomes. And all I need to do to add another reference is a little bit of upfront encoding work. That's exactly the kind of pivot to a novel technology and then scaling it that allows you to start to think about how would I detect pathogens that I don't know anything about. And now the unknown is not so unknown, and maybe you can catch the next pandemic threat. And I will point out that while we're doing this with SARS-CoV-2, because that's where we have good data, this approach works equally well when you apply it to the hantavirus stuff you've been hearing about, the Bundibugyo Ebola outbreak currently going on in the DRC. There are a number of emerging threats that we don't surveil and we don't look for because we don't have an easy way to see it. Here is a potential path to fixing that. So we do maybe have the technology to prevent the next pandemic. We are actively figuring out how we're going to scale this up. It really starts on the front end of this. We're going to need better sampling methods, and we are going to need better sequencing methods. So I told you a little bit of the surveillance strategy where we're looking at air sampling at large sporting events or pooled sampling of people coming off of planes, trying to just see is a virus circulating in the population and can we detect it there? The hope out of this is you give the public health community a head start on what should we be doing about the next virus rather than sitting on our hands and hoping that something will change, which was a lot of the way that the response to the SARS-CoV-2 outbreak went. We didn't have a lot of containment around it at the time. It was likely on US soil well in front of when it was pointed out in the media, and we really had no early defense against it. Nothing that we could do, nothing that we could change. Um, would that be better if we had seen it earlier? I certainly hope that the answer is yes, and that next time we will have a better response to those kinds of outbreaks. But that starts with, can I see it? Um, there is a follow-up here. You can go and see some of the extra information out of this presentation. I am curious, is there anybody in the audience that is actively using machine learning techniques or AI techniques to directly drive some of your work or are thinking about doing so? I'd be curious to take any questions. Because this is just me giving a talk this time, I'm also happy to end it a little bit early. And Adam and I won't banter back and forth the way we might on most other days. I'll pause there and leave just a little bit of space. Any questions? That's okay. If you do have questions, we're here. We'd love to talk to you. Um, I would encourage everyone who's attended to really be thinking about how you should be leveraging these newer generation of technologies in your work. They are transformative and they open pathways to kinds of research, to the next interesting move in figuring out how your business should operate that traditional software does not. Machine learning has been making an impact for decades now. AI, just in the last couple of years, and if you're really following it close, even in the last 6 months, has become massively transformative. If you are having trouble hosting those things, please come talk to us. We'd love to hear what you're up to. I'm going to go ahead and end us a little bit early today. Austin, any closing remarks for us?
Yeah, I want to start by saying thank you, James, for giving this talk for us. It was really nice. Thank you, everyone, for joining us. If you enjoyed this conversation, good news. We will be back in August to talk more about real-world ways to use AI in the lab. If you liked this talk or you're interested in hearing more, as always, please reach out. You can visit us at yaharasoftware.com. If you visit the About Us page, that's one of the easiest ways where you can get in contact with us. Otherwise, you can reach out to me or anyone really at the Yahara team, and we will find a way in which we can help with you getting AI off the ground in your lab. But until August, thank you guys for joining, and we'll see you next time.